Please do your homework, as you should always do before thinking about investing in Pressure BioSciences. We are a Boston-based company. We're about 30 miles south of Boston. We have a staff of 11. What I'm going to do today is give a brief overview of this company, and then I'm going to talk about some things that have happened in the last few months since I was here in December with a small group and also in January. We've got 24 patents in an area that we call pressure cycling technology, or PCT. Basically, our technology is a way in which we control molecular action. We can open up viruses and bacteria. We can kill. We can activate. We can control the mechanisms of molecules in a test tube by putting them under pressure. But not just pressure. Our patents are in turning pressure on and off, in cycling pressure. Pressure is a very, very powerful natural process, but it really has not been used that much in the laboratory. Every biological sample has a pressure point. Every cell, every molecule has a pressure point at which we can control it. We control molecules through heat, through cold, that is through temperature. We control molecules through chemicals, but we've never really taken advantage of controlling molecules through pressure, mainly because no one has ever been able to develop an instrument and consumables that could safely and exquisitely control pressure, particularly high pressure in the laboratory environment. Our pressures, if you think about the Marianas Trench, 6 miles down in the Pacific Rim, you're at 17,000 pounds per square inch if you're standing on the bottom of the Marianas Trench. We'll get 17,000 PSI, pounds per square inch, in a split second in our pressure chamber. We'll get 35, we'll get 50, we'll go up to 90,000 pounds per square inch. It's very safe, it's exquisitely controllable, and we're able to use this in an instrument that can sit on a laboratory benchtop and allow the laboratorian to access samples, parts of samples, proteins, lipids, DNA, RNA, to get them out in a way, as I said earlier, squeezing them out. It's like treating the cell like a sponge. Others, and I'm a scientist, for years, for generations, we've broken up samples by beating them up, mechanically beating them up, to open them up and get the 10,000 proteins liberated, as many as we can, and the lipids liberated. What we do is we squeeze them, we put them under pressure, and we cycle on and off, on and off, on and off. Once we find the pressure point to manipulate, to break it open, to inactivate, to stop the infectivity of an HIV virus, of a hepatitis virus, to break open a bacterium or a cancer cell or a normal cell. Once we find that, we'll put that into the computer and we'll be able to access that the same way every time, which is so important in science. So again, we have 24 patents that are controlling this process. We develop and sell instruments and consumables that use this process called PCT, pressure cycling technology. We've de-risked this investment. We have about 275 instruments in the world now in the field. All of these have been sold. 275 instruments that are being used by some of the top labs in the world to access the proteins and lipids and DNA and RNA in their sample. We have Merck, Lilly, Pfizer, Monsanto, Bristol-Myers Squibb, Amgen, Biogen, Harvard, Stanford, UCLA. We have the FBI, the FDA, the CDC. We have a number. We went after the key opinion leaders and asked them to look at this in the very beginning, and now we have over 100 publications that are out in peer-reviewed journals that are talking about the efficacy and the use of our technology. As a company, we have about 22 million shares outstanding, but if you look at the preferred shares, deals that I've done myself with about 200 high net worth individuals over the last 7 or 8 years, and you look at the 6.3 million we just raised in a very safe convertible debenture, then we're looking at about 60 million total shares outstanding. So the company has a market cap of about $30 million as we stand. We have revenue. In 2014, we did $1.3 million in revenue. 2015, we did $1.8 million. That's a number I couldn't give when I was here in January. And we are looking at giving guidance that we should be doing between $2 and $3 million in revenue this year. We expect this year's revenue to certainly significantly exceed last year's $1.8 million revenue for a number of reasons that I'll point out. I'll talk briefly about the group, terrific group. Dr. Ting, Dr. Lazarus, Dr. Lawrence have been with me for over 10 years now. It's a wonderful brain trust that I have some of the top high-pressure people and protein and DNA people that I've ever worked with. Dr. Ting is probably one of the top high-pressure chemists in the world, engineers in the world. I've started 3 prior companies worth $1 billion in market cap. Boston Biomedica in my garage grew to 300 people, 130 million, 140 million market cap. Changed the way that the world tests its blood for infectious diseases like HIV and hepatitis B. I spun out an anti-HIV drug discovery company. Panicos Pharmaceuticals went to 700 million market cap. And I've also was in the founding group of Trinity Biotech, which is about a 200 million market cap diagnostic company. It's still in existence. All 3 were NASDAQ companies. We are an OTCQB company. We're a fully reporting company. We've made it our goal this year. We had goals last year and this year. We've already succeeded in several of our goals this year. And the last one that we are looking at is to move the company back up to NASDAQ. So many of the things that we did last year and this year are on that path to move this company back to a NASDAQ listing. This is a cell. It can be any cell, animal, plant, human. This cell can be normal, precancerous, cancerous. This cell contains multiple proteins, maybe as many as 10,000 different proteins, maybe as many as 1,000 different types of lipids. And of course, inside we know it's got nucleic acid, RNA, and DNA. The whole purpose of a scientist now in the biological area is to break this open. We don't look under microscopes that much anymore. We are at the molecular age. We're looking at not just proteins, but we're looking at the building blocks of proteins. We're looking at peptides that make proteins. We're looking at amino acids that make peptides. We have to break these out, and we have to find one protein in this gamache of maybe 10,000 proteins that we think is related to a disease or can be part of a therapy. We have to pull that protein out. We have to break it down into its building blocks, which are thousands of amino acids, and try to see if one change might be the change that is causing a certain disease to be prevalent or certain disease to be very progressive. Our job at Pressure BioSciences is to offer an enabling technology to the scientists to allow them a better way, a more exquisitely controlled way to break open the sample and to get to those biomolecules that may in fact be the next billion-dollar drug. This is a million-dollar machine called a mass spectrometer. There are 16,000 laboratories around the world that have one or more mass spectrometers. That machine will exquisitely read proteins that you put into it, and it'll give you that readout. But you can't put a whole protein in. You need to break open a cell, pull out the protein of interest, break it down, and then put that into the machine. On the left, that's where we believe we should be. 16,000 labs with one or more mass spectrometers. We believe we should be in front of all of those. We believe we do a better job of breaking open and breaking down the samples than what's out there now. How do we do it? We set a goal about 3 years ago to work with one of the big 6 companies that makes the $1 million mass spectrometer. I say $1 million. They run from $500,000 to about $1.5 million. And that was one of the goals that we had, which we accomplished earlier this year in January. So what is the value proposition? The analysis of the RNA, DNA, protein, and lipids is absolutely crucial to finding the biomarkers that we're looking for. But the problem we have is accessing these from the cell is a very, very difficult problem. It is highly inadequate. It is a bottleneck. Talk to any researcher that's here. I see some in the room here, or anyone you know, and they'll tell you the machines themselves are terrific. The data reduction is terrific, but accessing, breaking apart the cells is the key. Sample preparation is the key, and that's what we do, and we think we do better than most. It's a very large market. It's a multi-billion-dollar market. As I said, we have a number of researchers that are out there and some very well-known labs that are already using it, already publishing. This is a picture of our instrument. You can see it on the bottom. It has a pressure chamber. The chamber is about 1 inch thick steel. We fill it with water. We compress the water. Water compresses about 6%, so it's not going to blow up. We put our samples in special consumables, and the cells go into a test tube, fill that with buffer, put that inside the water chamber, close it, and then the computer, you say, how much pressure? How long do I want to put it under pressure, and how many times do I want to squeeze it back and forth? Turn it on and turn it off. And once you do that, it does the work for you. At the end, you've broken up your cell, you've liberated all those biomolecules that you need to do your research. So we sell the instrument. We now have 5 instruments. We're excited because we have 2 new instruments that we've worked on and we've released. Now we're ready to sell them this year. And we have consumables that go along with everything. There's 8 areas. I can't go through them, but I can tell you that the USDA and a number of medical schools around the country are using this to look at areas like food science, antibioterror, drug design. They're using this to break open the samples, whether it's in agriculture or aquaculture or human disease, to free and liberate those biomolecules. This is Dr. Rudy Aebersold. He's the guy that put us on the map. He's one of the world's premier protein chemists. He is a keynote speaker at many, many protein meetings. And Dr. Aebersold published last June a paper in a major journal called Nature Medicine, and said, in his opinion, if you use pressure cycling technology up front to prepare your samples, and you use mass spectrometry, particularly the mass spectrometer from a company called citing, which is the largest company in the world selling mass spectrometers for protein analysis, he thinks that you would have the best chance of getting your best results. We make PCT. citing makes what's called the SWATH, S-W-A-T-H, mass spectrometer. He called his method PCT SWATH. So as I said, one of our goals last year was to do a deal with one of the 6 major mass spectrometry companies, Thermo Fisher, Agilent, Bruker, Shimadzu, Waters, and citing. citing is owned by Danaher. Danaher is a $68 billion holding company in the life sciences area. So once this paper came out, naturally my first call was to the people I knew at citing and said, is it time we now really talk? And they said, yes, it is. Very difficult as a company of 9 people at the time, living on the edge, living paycheck to paycheck for 4 years, having a disruptive technology that not everybody really has bought into or really even understands, talking about taking pressure that's equal to 3, 4, 5 times the Marianas Trench and putting it on the lab bench. Very difficult to bring that to one of these big companies and say, hey, you want to do a deal. But when Professor Aebersold comes out and talks about this is what we use and this is why we use it, it opened up the door. And so, in fact, on January 12th, this year, citing announced that they have signed a 2-year exclusive co-marketing deal with us. This is David Weil. David Weil was a former vice chairman of NASDAQ. He's given me permission. He said, use my picture, use a slide, let people know that even though I ran NASDAQ for a number of years, you are the only OTC company that we've ever invested in. So David himself personally and his group invested in Pressure BioSciences last year and said, we're going to help you get back on NASDAQ because this is where your technology company belongs. Key achievements in 2015. We are awarded a $1 million grant. Those of you who were in the audience in January saw these. Just a reminder, we developed and we have a patent pending on a very interesting consumable that Dr. Aebersold has talked about, thinking that this might be the way when you do biopsy samples, whether it's liver or whatever, when you do a biopsy sample, nowadays they're doing what's called small needle biopsies, very small pieces of tissue, not big chunks of tissue. They usually take multiple sites, maybe 10 or 12 sites with a needle biopsy. It's so small that they cannot run mass spectrometry on that biopsy tissue. He's found a way using our method and this new consumable to run mass spectrometry. The current methods get you 100 or 200 proteins from that biopsy tissue. Our method, according to Dr. Aebersold, gets you between 2,000 and 3,000 proteins. And he's written this up, and several other groups have written this up in the meantime. Dr. McCord is a forensics expert, and he's working on an improved rape kit test. It's a national travesty. There's 400,000 untested rape kits in this country sitting on shelves. The main reason, it's a very expensive test and a very time-consuming test. What if you can do this test much faster? Maybe we can start bringing this backlog back down to and getting rid of the backlog. And it takes an entire day, the first day, to separate from a vaginal smear the perpetrator sperm cells from the female epithelial cells. Both contain DNA. So you have this cotton swab. You have to spend an entire day to separate, and then you break those 2 cells and free the DNA. You cannot mix them. What if you could put that into our tube, into our pressure chamber, dial in a pressure, and break only one, like the sperm cells, and not break the female epithelial cell. You can do in minutes what now takes an entire day. And this is what Dr. McCord is working on. We're working very closely with him. We're hoping that by the end of this year, we will have something that's much closer to a commercializable test. There's a number of papers that have come out last year that talk about the technology we have. We have collaborations with one of the most prestigious research organizations in Italy. So we have a number of great collaborations going on. We started last year planning our uplist to NASDAQ. We sold our stock to Everest Investments. We have a very tight relationship with a number of financial people, particularly fund managers in Poland. More to come on that as the weeks and months go on. But Everest Investment is run by one of Poland's most respected fund managers, and he has 1 million shares of our stock, and we're hoping that as his fund grows, that is going to grow. And our revenue in 2015 exceeded 2014 by over 30%. 2016, we closed a $5 million pipe at $6.3 million. We actually desired to go to about $7.5 million, but because of legalities, we had to close it down at $6.3 million. So those of you who have time to meet with me afterwards or have a one-on-one, I'd like to talk to you one-on-one about what we're doing going forward, but we're very happy that we were oversubscribed for a $5 million pipe. It was a convertible debenture at a set price at 28 cents, and we started it when the stock was at 20 cents and raised half the money when the stock was under 28 cents and the other half when it went over 28 cents. We got rid of all of this ugly, toxic, variable rate debt. It's absolutely gone. Nobody ever converted. It's a very risky thing to do, but we grew our company with this variable rate debt. It's now gone off the balance sheet. We have a co-marketing partner with Cix, which is wholly owned by the Danaher Company, this $60 billion company on New York Stock Exchange. We were added 2 weeks ago to the high-performing Richmond Club Index in Toronto, and we're very proud of that. Richmond Index has outperformed the S&P by about 17% for 10 years in a row. We were added to the index 2 weeks ago. We talked about PCT swath and a number of things that are going with Dr. Aebersold, and we continue this year right now our uplisting expectation for NASDAQ. We've hired two of the best in the country that we believe to help us get there, and our price has increased so far this year from when I was here 3 months ago at 32 cents to about 47 cents in April. This is a VWAP. We did hit in the high 50s. We've been in the 50s now for about a week. Nice chart. Sometimes when I'm lying upside down in bed, I see it, and it's not this way, but I'm looking at this saying, hey, this one is looking good. This chart is finally looking good. It's a very slow growth. We're doing this the right way. We're doing it by getting out and telling everybody the story, and by having our sales increase, by doing deals with companies like Cix, by cleaning up our balance sheet, and by talking about our desire to get back on NASDAQ. I've only run NASDAQ companies. Running an OTC company is very, very, very difficult. As I said, our volume in shares has been increasing. The turnover every day, the price of the stock's been increasing. The number of trades has pretty much been increasing since I've been here in January. A couple more slides. The Children's Hospital Medical Research Institute in Sydney has got a $10 million grant. They're going to test 70,000 tumor samples. They decided that they're going to use PCT swath. They'll be ordering 3 of our instruments, which we expect to ship them in June. We've already received the purchase order. This is the first order with our deal with Cix. So we're very happy with this, and we think this is one, and Cix thinks this is just one of many laboratories that are going to be doing what they call industrialized proteomics. I do not have time for this other than the key point here is if there's 16,000 labs, individual labs have one or more mass spectrometers, if we can get 25% market share over 5 years. And I think that's a very, very reachable goal for a company that has the next best thing since sliced bread, in our opinion. That's going to give us $75 million in instrument sales, but a recurring revenue of $36 million a year. Remember, razor blade story. You buy the instrument, It's a one-time purchase, but then you've got to use, every sample you use has got to use its own consumable. Our short-term drivers here, we have this major deal that we just signed. It's going to take time to get it going, but time means, in my opinion, this year. We've already talked about the group in Sydney. There are other groups we're talking to, so it's not going to happen overnight, but it's great to have a co-marketing partner that has access to distribution network through their own distributors, their own salespeople in over 100 countries. We have one salesperson in the entire company. So our $1.8 million last year was from one salesperson. We've cleaned up our balance sheet. We are going to be adding between 2 and 4 salespeople this year to help augment everything. We plan, as we say, to uplist. We've got a new powerful instrument that is going to be available for the SciEx people and then for everybody starting in June. We've been working on this for over a year. We have wonderful collaborations that I've mentioned about, and we think that the continued sales increases, our new product releases, our additions to our sales staff of 1 to bring it up to 4 or 5, and our healthier financial position and the SciEx co-marketing deal are all going to add to our success that we look forward to in 2016. Thank you very much.